Okay, so this is the second prep for lecture 21. And I just wanted to start with this. This just shows a number of different maps that have been obtained from different HFR strains. And when you first look at these, there's the H strain and the 312 and the 2 and so on. You look at it, it seems kind of random, right? It doesn't look like there's any coherence. But if you remember, if you remember that the bacterial chromosome, there's a number of different strains where the F factor has inserted into different locations and in different orientations, so that, if you do a mapping experiment, you can, detect these genes in this order, or these genes in this order, or if the F factor's in a different orientation, these genes in these orders, and so on. And that by putting together the information from a number of different crosses, you have a picture of the bacterial chromosome. And so just to point out, if you just take a reference point, so let's just take the glycine gene, you will see that it's present on all of these different strains, of course, because they make their own glycine. And if you look at the genes next door, so the one next to it is histidine, and then gal, there's that noise again. It didn't make any noise the whole time in between these recordings. It's very interesting. And then there's his and gal, right? His and gal, etc. So if you look at those, you can see that the gene orders haven't changed and the distance of parts haven't changed. It's just where you start and end your map has changed. And this is a very important symbol here. This is the fertility factor. So if you saw this in a bacterial chromosome, sorry about the terrible drawing, but if it was like this with this little arrow this way, and then this part here, this arrow, this is the DNA that is first broken and then fed into the recipient cell, and the genes, these genes are fed in, and then these, and so on. And if it went the whole time, eventually what would happen is the terminus would enter. The terminus is the part of the F factor that contains the genes that are needed for producing a pilus and being a donor cell, and so on, and initiating the transfer of the DNA into the recipient cell. And so when you see this, that's fertility factor, but you know that the genes on this side of it are the ones that are going to be added in an experiment, are going to be entering the recipient cell, and that this is kind of the end point. So if the whole chromosome entered, which it almost never does, this arrow indicates, the proline gene is right next to it, and then the threonine, then thymine, and so on. And the terminus is right after the lac gene, and so on. So notice a couple of main points here is, first of all, that symbol, because if you're given an image and asked which the first and last genes will be to be transferred, it's important for you to know which is which. And also just that these genes they don't change their position or location or distance apart from one strain of E. coli to another, but the different strains just have that F factor in a different location. And so this shows you the circular sketch of the chromosome for the H strain, strain 1, strain 2, and so on. And the 3 and 312 are on the other one, okay? So this is, it's because of this that you can actually use that consistency in the gene order and approximate distance to do experiments like interrupted mating that allow you to figure out the relative position of each gene. If I'm studying this particular strain and I find out that purine and lactose, the lac genes are like 3 minutes apart, and these guys are 16 minutes apart, and these are 12 minutes apart. They will be the same distances apart in all the other experiments as well. So the distance, the timing doesn't change, just where you start and end, I guess, is the most important. Okay. And when you're given questions to answer, I'm just digging around my old files just to see if I can modify some questions for practice for the next PEQs. And the chromosomes are circular, so there's no sort of ends to the chromosome the way there are with eukaryote chromosomes. So generally what I will say is, list the genes in order, starting with lac and ending with, whatever, so that you have some sense of what order to write them in. Okay, and just again, to make the point, there are 2 different experiments indicated here. There's this one where the D, C, E, A, and B genes were mapped. And then here's a second one where the H, E, G, B, F genes are mapped. And so what you find out for E and F are consistent between the 2. That's why these 2 lines are here. So we know that between the E and the B gene, we have 12 map units, right? And we know that between E and A is 6 minutes, not map units, and E and G is 3. And so we can deduce that there's a 3-minute distance between the A and the G. So when you put these maps together and you line them up by including, the ones that are the same for both, like we could have, well, no, we couldn't. Then you have reference points, and then you can kind of map out from the distance. So the distance between E and C here is 12 map units. Between E and H here is 10. So we know that's a 10 map unit distance, and we know that's, sorry, I keep saying map unit, minute, a 2-minute distance, and then we know this is 8 minutes because of this information. And if we had to deduce, we could say, okay, well, the D and the E gene are 20 minutes apart, right? So you could also be given information like this and asked to produce something like that, right? It would, on an exam, it would have to have, I think, fewer reference points than this, because you want to make sure people have enough time. But because the gene orders and distances are consistent from one type of bacteria to another, from one strain to another, you can overlay the 2 experiments and then get much more detailed information than from one or the other. We have more information in our composite here than we do of each of the 2 individual ones. And so, this is just another example. The orange one on the outside shows you the overall map. And then the different colored bands here are just different experiments that were done. And it shows you how, we get sort of the information. And when you overlay them, you get all the genes, right? This purple experiment didn't include the A gene, but because it included B and G, and we know that information, then the BG distance and the BA distance can be determined, and then the AG, which is the one we don't have, right? So that's the kind of stuff that we can put together. In this experiment, the H gene wasn't included, but we can figure out where it is based on the overlay from these other 2. So from the green one, we do know where H is and C, and so we can, extend that this way. Yeah, and so this is just an example of how many different experiments are put together to get a full comprehensive map. Okay, and so the kinds of crosses that can be made, you have to have a donor cell, which is either an HFR, remember that's the one with the F factor incorporated right into the chromosome, or F plus. And then you have to have a recipient, which is your F minus cell. So this is a cross that can take place, and so is this, but none of these others can happen. You've got to have one donor and one recipient, so you can't cross F minus cells or F plus cells to each other, or HFRs to each other, or HFR to F plus, and so And just the genetic map we generate from interrupted mating is a crude map. However, it's a useful starting point because if we know the relative order of the genes and just a sense of how far apart they are, that helps us to do a really good design of the crosses we want to do HFR mapping, which requires a lot more care and attention. You're going to look at lots of different cells, colonies, determine their genotype. It's a huge amount of work. So the more information you have to start with, the more likely it is that your experiment will work well. And we're going to talk about HFR mapping exclusively in the final lecture, lecture 22. Okay. Now there's another thing you can do to get a sense of the distance two genes are apart on a chromosome. It doesn't tell you sort of order because we're generally doing just 2 genes at a time. So gene order is not really relevant, although you could overlay several co-transformation experiments and get a sense of gene order. We're not going to go there. So this is just a reminder, if you go back to those Griffiths experiments where they determined that DNA was over time, they determined that DNA was the genetic material, it started with a transformation experiment. So we had live R strain, which was not virulent, and we had heat-killed S strain that had been boiled or autoclaved or something, and neither of those could cause infection on their own. Because the S strain, the DNA was present in the tissue, but you can't give mice bacterial DNA and cause disease, right? It has to be inside a bacterial cell. That's how the disease is actually brought into the organism. But when you mix that R strain and that S heat-killed extract, some of the R cells were able to take up DNA from the killed S strain and became S strain cells themselves. And so so this was referred to transformation, and we still call it transformation. And the reason is because the R strain was transformed into a virulent S strain. So the way we do these kinds of experiments, if you go on to take genetic engineering and some of the MBB type courses, you'll learn a lot more about this process, about exactly how it's done. The details. But the idea is you take bacteria that are capable of taking up the DNA from the environment. Not all of them are. It depends on the type of bacteria, and it depends on some other conditions which you will learn about in other courses. But the idea is that you isolate DNA, and then you break it up to pieces that are they may vary in size a little bit, but on average they're a certain size, 5,000 bases or 2,000 bases or something like that. And then you expose them to bacteria, and you provide the conditions where the bacteria could take up that DNA. And you give them a certain amount of time to do it, and then we basically look for cells. We plate selectively to collect cells that have taken up some DNA, and then determine whether the genes we're interested in. Suppose we're studying 2 genes, and maybe they are pretty close together on the chromosome. And the genotype of the bacteria is A minus, B minus, and then we do the transformation experiment, and then we test to find out how many of the cells took up the A plus gene, how many took up the B plus gene, and how many took up both genes. And if a lot of cells take up both, then we know that the genes are pretty close together on the same chromosome, because they must have been taken up on the same piece of DNA. And so that's called co-transformation frequency. So the frequency at which both of the genes are taken up, excuse me, compared to just one or just the other, is what tells us the relative distance the genes are apart. So for example, suppose we had a gene for production of histidine and production of arginine, and it turns out that they're 20,000 bases apart on the bacterial chromosome. And our recipient bacteria is an auxotroph, and we take DNA isolated from another strain that's prototrophic, And we break up the DNA. There's different ways you do it. You can sonicate it, you can actually autoclave it, or run it, suck it up into a very fine needle and push it back out into, and sort of push it back out a few times. That can shear it. And let's say our average size is about 8,000 bases. Okay, and then you basically collect some cells after the process is done. Say you select 500 colonies, you plate them on full media so they all can be all possible genotypes, and then you plate them on media just lacking the histidine, and you get 50 colonies. Okay, so that's the transformation 50 over, I made the numbers easy so that you could do them in your head. It's 0.1 transformation efficiency. And then suppose you do the same, you replica plate them on media that's lacking arginine, 45 colonies, so that's 0.09. So the frequency of one gene being taken up is about 0.1. And then the frequency of the other gene taken up is 0.09. And so think about this for the next recording. What is the chance that we get both histidine and arginine genes in the cells if the pieces of DNA that we're putting in are about 8,000 bases long, but the genes are actually around 20,000 bases apart? Okay. And you need both of these frequencies to try and figure that out. And we'll start with that on the next video.